So innate immune system consists of a cellular and of a humoral arm. And here is where all the hype and attention is, namely cellular sensors of tissue damage and microbial invasion. But the innate immune system also includes a humoral arm, which is frequently depicted as a collection of weird molecules. So I will use one of these molecules, which we called pentraxin-3, as a tool to understand the interplay between cellular and humoral innate immunity. And in the second part of the talk, I will try and share with you some unpublished data and concepts. So if innate immunity is like speeding, it's a car that speeds. This is Umberto Boccioni. And if you speed or drive a Ferrari, I drive a Vespa, but if you drive a Ferrari, you need brakes. And we have long been interested in brakes. We have long been interested in the IL-1 system, and many years ago we described the type 2 IL-1 receptor as a decoy, which in membrane-bound and released form acts as a molecular trap of the agonist, and this molecule also acts as a dominant negative. And ever since, we have been obsessed with decoys. And unfortunately, this is not an Italian wine. It is a Californian wine. But there are several bottles, and we have contributed to a couple of these bottles. But it turns out that decoy receptors are a general strategy conserved in evolution to tune the action of inflammatory cytokines, chemokines, and growth factors. In addition, again, given our obsession with negative regulation and decoys, many years ago, Marta Muzio and Nadia Polentarutti in my lab cloned a member of the IL-1 receptor family, which at the time we called T-rate because it was the 8th molecule. Now there are many more with a TIR domain. And it looked like the fringe member of the family with a long cytoplasmic tail and 2 amino acid substitutions, which suggested to us that it may not be a conventional signaling molecule. And work done by us and by others, in particular by Shi-Xiao Li in the United States, suggests that indeed this molecule is recruited at signaling receptor complexes of the IL-1 and toll-like receptor family and dampens signaling and inflammation. And I want to convey a general message about these brakes. And the general message, and this general message is drawn based on work that we have done on 2 negative regulators, namely D6 in the chemokine world and T-rate in the IL-1 receptor world, so different structures, different ligands. And the general message is that these negative regulators serve to tune and strike a balance between the amplification of innate resistance and uncontrolled inflammation. Moreover, they are essential for resolution and repair. And dampening inflammation and resolution and repair will be a recurrent motif in the rest of my talk. So yesterday we were confronted with old technology, and Tim Mossman mentioned the Mtt assay. And years ago we were using old technology, which is shown in the next slide. Again, we were interested in the IL-1 field, and using old technology, we were looking for IL-1 inducible genes. And this is the technology. We were fishing for IL-1 inducible genes in endothelial cells and macrophages. And one of the fish that we caught, we called pentraxin-3 or PTX3. We cloned the mouse and the human cDNA and genomic. It is a secreted molecule consisting of 2 domains, a pentraxin domain coupled with an unrelated N-terminal portion. The pentraxin domain most likely with a jelly roll type of topology. The molecules assembles as an octamer with intra and inter-chain disulfide bridges being essential for the structure of the molecule. And this is work done mainly by Antonio Inforzato in our laboratory and collaborating with Tony Day in UK. This molecule is related to molecules which form pentamers, and the classic one is C-Reactive Protein, and the other one is serum amyloid P component. And it is my tenet and belief that indeed C-Reactive Protein, which was identified by Avery and colleagues and by Tillett and Francis, indeed is the first pattern recognition molecule to be identified with antibody-like, and I stress this point, antibody-like properties, being able to recognize the structure of pneumococcus. We were lucky because we cloned the first member of a family, what we call the long pentraxin family. These are molecules which we cloned two of them, PTX3 and PTX4. These are molecules are made of a pentraxin domain coupled with an unrelated N-terminal portion. The molecule that we cloned, PTX3, is strictly conserved in evolution. Unlike C-reactive protein, whose structure, regulation, and ligand recognition is not conserved in evolution. So we do not have genetic evidence as to the function of C-reactive protein in spite of its widespread use as a diagnostic, as Charles Dinarello mentioned today. So we have been using this molecule as a paradigm for humoral innate immunity to understand its relationship to the cellular arm of innate immunity. This slide also emphasizes that pentraxins are conserved in evolution. Here is toru fish, somewhere here is Drosophila. So from an innate immunity point of view, these are respectable molecules conserved in evolution. Many cell types are able to produce PTX3 when confronted with inflammatory signals, be it TLR agonist or primary inflammatory cytokines. On top of the producers are macrophages and myeloid dendritic cells. It is not in the repertoire of plasmacytoid dendritic cells. In terms of regulation, it is production is co-stimulated by IL-10. This is work that was done years ago by Andrea Doni in my laboratory. So the molecule pops up in profiles of what we call M2-like cells, just to emphasize some relation with the classic alternatively activated macrophages, M2 macrophages, which were described in a seminal JXMed paper by Simon Gordon and colleagues. In addition, a few years ago, and when I say we, it was Giuseppe Peri in my lab and Sébastien Jaillon in the lab of Pascal Janin in France. So we, and I say we because instead of competing, we published the paper together. We observed that PTX3 localizes in neutrophil granules, and upon extrusion of neutrophil granules and formation of neutrophil extracellular traps, these are structures that were originally described by Arturo Ziglinski in Berlin, PTX3 associates with nets, and nets serve as focal points to favor the interaction of antimicrobial molecules such as PTX3 with microbes. So neutrophils indeed are literate cells. They have a complex vocabulary as shown here, and P for us in terms of neutrophil stands for PTX3. So what does the molecule do? Well, first of all, it recognizes microbes. Oops. Well, first of all, it recognizes microbes, and the first ligand we identified is outer membrane protein. Outer membrane protein A is a structure which is strictly conserved among enterobacteria, and it is essential for the viability and pathogenicity of enterobacteria. So it fulfills the classic criteria that were originally postulated by Charlie Janeway. Recently, and this is worked from bed to bench, we identified other microbial structures recognized by PTX3. And this study was prompted by an observation. This is work done in collaboration with Sprong et al. In meningococcal sepsis. And here are very high levels of PTX3, which is an early marker, and this is a general rule, an early marker of inflammation. In many cases, better prognostic marker than C-reactive protein. So these clinical observations raise the issue of what is the molecule doing in meningococcal infection. So, and this is work done by Marina Sironi and Barbara Bottazzi in collaboration with Rino Rappuoli in Siena and his colleague Pizza. And in essence, we found that the molecule binds meningococcus, and thanks to the library of meningococcus B molecules that they provided us, we identified three molecules which are recognized by PTX3. I won't dwell into the details, but in essence, PTX3 binds a capsular Neisseria meningitidis, it binds outer membrane vesicles, and we identified three meningococcal proteins, again proteins, much to our surprise, which fulfill the classic criteria for PAMP. In addition, and this is work that we did, actually they did, it's Joe Teti who did the work and these people in Messina, exogenous administration of PTX3 reduces the bacterial burden in a model of infection. These are neonatal infection. And PTX3 deficient mice show lower antibody titers when challenged with outer membrane protein, and this is rescued by exogenous PTX3. So our working hypothesis, and we are still working on this, is that PTX3 acts as an amplifier, if you pass me the word, as an endogenous adjuvant in the adaptive response elicited by outer membrane vesicles of meningococcus. PTX3 binds complement component, and actually this is the original experiment done by Barbara Bottazzi, who originally observed that PTX3 binds C1q. So my reaction when I saw that piece of data is here, complement no, and I apologize with my complement students, friends. But we were lucky because we talked to the complement people and collaborated originally with Modaha and Franco Tedesco, and more recently with Seppo Meri, Ljubka Rumenina, and Peter Garrett and Nicole Thielens. And this is the picture. The molecules interact with C1q, activates the classic pathway of complement activation. In addition, it interacts with factor H, and here we collaborated with Seppo Meri. So alternative pathway. In addition, it interacts with other humoral fluid phase pattern recognition molecules, including ficolins, and this was originally picked up by Andrea Doni in an effort that we did to define the interactome of PTX3 and MBL. And this has implication for cooperative microbial recognition and for the logic of the function of the humoral arm of innate immunity. So this is a summary, again, the gene, the molecule, the structure. We have seen the molecule, which was a great emotion. It binds selected microbial components, and I showed you some unpublished data on that, microbe recognition. It interacts with Fc gamma receptor, the work of Lu et al. And the work of Cecilia Garlanda and others in my lab. So opsonic activity. It interacts with components of the complement cascade. It interacts with other pattern recognition molecules, in particular some which have a fibrinogen domain, and I will get back to that. And this has implication for repertoire and synergism. And finally, and this is work of Libia Deban, a talented PhD student from Croatia, and again, in the context of a comprehensive interactome analysis of the molecule, she found that it interacts with P-selectin and regulates leukocyte recruitment and inflammation. So it acts as a negative regulator. And the genetic evidence shows that the molecule is essential for resistance against selected microbes, in particular Aspergillus fumigatus. Originally, we collaborated with Luigina Romani, and it is a regulator of inflammation. So I want to share some now unpublished, fresh new data, and at the end some ideas. Well, first of all, the first piece of data is an idea that belongs to Sébastien Jaillon, a talented postdoc from France. And Sébastien asked the question whether PTX3 is relevant for defense in the urinary tract. And this is a peculiar tract with peculiar pathways of regulation. It's a mucosal immunity tract with peculiar pattern of regulation, which we really understand to a limited extent. In addition, urinary tract infection is an important clinical challenge. So these are knockout mice challenged intrauterally, and this is again the talent of Sébastien, with uropathogenic E. coli and PTX3 deficient mice showing phenotypes in terms of increased susceptibility to uropathogenic infection by E. coli. And this is also reflected in terms of weight loss, and this has probably something to do with regulation of inflammation by this molecule. The abscess formation is dramatically different in PTX3 competent and PTX3 incompetent mice, and these are kidney abscess, so ascending infection. Which are the sources of PTX3? I will not belabor the point, but PTX3 comes, of course, from leukocytes. It's carried by neutrophils, but in particular, epithelial cells are really the director of the orchestra, we feel, and they are the main source of PTX3 under these conditions, we feel. The pathway is MYD88 dependent, it's IL-1 receptor independent. So most likely is a TLR dependent, primarily TLR dependent pathway of induction. The molecule has opsonic activity against uropathogenic E. coli, is shown, for instance, by exogenous addition of the molecule, which at very low concentrations, nanogram per ml, has opsonic activity. In addition, PTX3 deficient neutrophils have delayed and reduced capacity to handle uropathogenic E. coli. So PTX3 present in neutrophil granules helps the opsonization and the handling of the bugs. And in the absence of PTX3, there is uncontrolled inflammation and tissue damage. So this is a summary. PTX3 deficiency is associated with increased susceptibility to urinary tract infection and to uncontrolled inflammation. The PTX3 opsonized E. coli is more viable for handling by neutrophils, and in particular, epithelial cell-derived PTX3 contributes to resistance against urinary tract infection. And we looked for genetic evidence in humans, and we were lucky to collaborate with Katarina Svanborg in Sweden, who has she has very carefully collected case lists, and indeed, PTX3 genetic polymorphisms are associated to susceptibility to urinary tract infection in humans. So the main general message is that PTX3 is a key component of the humoral arm of innate immunity, and it is essential, non-redundant for resistance against urinary tract infection. And in the urinary tract, the cellular and the humoral arm of innate immunity exert complementary function. And this is schematic representation of this PTX3 downstream of the MYD88 pathway. It is produced at low levels under homeostatic conditions. Production is induced by sensing microbes. The molecule facilitates handling of bugs, in this particular case E. coli, by neutrophils, and finally it has a regulatory function on inflammation. As Simon was kind enough to mention, we have a long-standing interest in inflammation associated with cancer. And work that we have done and work that others have done have shown that PTX3 pops up in the microenvironment of some tumors. There is, for instance, a recent paper showing correlative evidence that PTX3 may be prognostic in carcinoma of the pancreas. So the question that we have and we had is, what is it doing there? And of course, Hal Dvorak many years ago pointed out in a seminal paper in New England Journal that tumors are wounds that do not heal. And so in parallel, Andrea Doni started a project on PTX3 and wound healing, and Eduardo Bonavida started a project on PTX3 in carcinogenesis. And we have interesting results in both. I will not mention the cancer data. I will just focus on the wound healing data, because hopefully it conveys a general message in terms of the mode of action of immunity, and the humoral arm of immunity. So after wound healing, and here we are dealing to skin wound healing, but Andrea has repeated the experiments, or the key experiments, also in a model of liver inflammation and in a model of lung inflammation. So after skin wound healing, PTX3 is induced. The induction is MYD88 dependent and only in part IL-1 receptor dependent. So presumably IL-1 is an amplification loop here. PTX3 is localized in the clot, and it is associates with the bundles of invading cells, in particular for, of course, dear to our heart, CD68 positive macrophages. I will get back to macrophages, but also to PDGF receptor positive, presumably cells of mesenchymal origin, which organize, Andrea made this observation, which organize into bundles. So what about the PTX3 deficient mice where there is a phenotype? PTX3 deficient mice show premature closure of the wound. They show a thicker clot at the beginning, increased fibrin deposition, and eventually a thicker scar with hyperplastic epithelium, and here you see lack of appendages of the skin. So there is a clear, fairly dramatic phenotype. The phenotype is rescued by exogenous administration of PTX3, and here, for instance, if you look at day 7, it's pretty obvious that the phenotype is rescued. And if you substantiate the observation of thicker scars by measuring collagen, indeed, the PTX3 deficient mice increased levels of collagen and increased level of collagen by histology by staining with Sirius red. So what is the mechanism? We focused on the hemostatic system for several reasons. First of all, it was obvious that there is a bigger clot, that there is increased fibrin deposition. In addition, we reminded ourselves of the fact that PTX3 interacts with pattern recognition molecules which are characterized by a fibrinogen domain. So Andrea found that indeed, among the various and complex elements of the coagulation cascade, PTX3 interacts with respectable affinity with two components of the coagulation cascade, namely fibrinogen/fibrin and plasminogen. But interestingly enough, the interaction is dramatically influenced by pH. The interaction is much stronger at low pH. And he has also done surface plasmon resonance and all the details. Is it functional relevant? Yes, because if we add activators of plasminogen, UPA or TPA, and add in PTX3 in vitro, and we allow a tripartite interaction, then there is increased fibrin degradation. I should mention also that we mapped the domain that is involved. It is not the pentraxin domain, but the N-terminal X domain encoded by the second exon fully accounts for recognition of these components of the coagulation cascade, fibrin/fibrinogen and plasminogen, and for the in vivo phenotype, because the phenotype is rescued by exogenous administration of the N domain, but not of the pentraxin C-terminal domain. Of course, macrophages are dear to our heart, and the molecule is produced by macrophages. And so Andrea looked in an in vitro model of fibrinolysis in which macrophages are put in a fibrin gel, and then we're looking for fibrinolysis. And as you can see, the PTX3 deficient mice show a defective capacity to mediate pericellular fibrinolysis. And this is reflected again, once more in vivo, measuring D-dimer as an expression of fibrinolytic activity in vivo. The PTX3 deficient mice show a defective production of D-dimers. And in addition, using confocal and two-focal microscopy, Andrea also showed that PTX3 and fibrin and plasminogen engage in a tripartite interaction in vivo. What about pH? Andrea set up a model to image pH levels in wounds, and there is a correlation. If you look, where it is darker, it's more acidic, and as you can see, there is co-localization of PTX3, fibrin, and plasminogen, preferential co-localization of these three elements in the areas which are more acidic. Okay. So, what is the evidence? At that point, the question is there actual evidence that the coagulation cascade is responsible for the phenotype? And to address this question, we either blocked coagulation or depleted mice of fibrinogen using antithrombin 3, argatroban, or batroxapine. And indeed, as expected, if you block coagulation or deplete mice of fibrinogen, you have delayed wound healing. And under those conditions, there is no phenotype of PTX3 deficient mice. And I think that suggests that the coagulation system is indeed essential for this phenotype of PTX3 deficiency. So this is the summary. Following wounding, PTX3 is associated with thicker clots, with increased fibrin accumulation, and this is followed by thicker collagen-rich scars. I should emphasize that the fibrin mesh provides a provisional extracellular matrix essential for the proper organization of the final extracellular matrix and tissue repair. Exogenous administration of PTX3 reversed the phenotype, and I should add that the N-terminal domain encoded by the second exon is fully responsible for this function. Moreover, a similar phenotype was observed in models of liver and lung inflammation and tissue damage, and more recently in a model of thrombus formation. The interaction of PTX3 occurs preferentially, not exclusively, but preferentially at an acidic pH, and using in vitro and in vivo systems and cellular models, this tripartite interaction promotes fibrin degradation. And again, timely fibrin degradation is essential and heralds appropriate repair. And by interacting with the positional matrix protein fibrin and plasminogen, PTX3 plays an essential role in the orchestration of tissue repair and remodeling. And as a consequence of metabolic adaptation, we all know that an acidic pH occurs at sites of wound repair and in tumors, I would add, and a low pH acts as a switch-on signal for this function of PTX3. And this is represented in a schematic way here. Here are cells of the monocyte macrophage lineage and neutrophils producing PTX3 at neutral and acidic pH. And this again is work of Andrea. There is recognition of microbial moieties, activation of complement components, opsonic activity via Fc gamma receptor, regulation of inflammation via P-selectin. Acidic pH acts as a switch-on signal, and we have the formal proof during wound healing, and sets the molecule in a tissue repair mode by allowing and favoring its interaction with fibrin and plasminogen, and this is essential for appropriate resolution of inflammation and orchestration of tissue repair. I want to share some speculation and views. I think it has long been known that selected components of the extracellular matrix, including fibronectin, vitronectin, osteopontin, and mindin, have opsonic activity. In addition, some components of humoral innate immunity, these humoral fluid phase pattern recognition receptors, and I showed you the case of PTX3, but this is also true for other, such as serum amyloid P components, C-Reactive Protein, serum Q, and collectins. Now, these molecules interact with extracellular matrix components, and in addition, some of them have extracellular matrix modules, such as the fibrinogen module in fecolins. And there is evidence suggesting evolutionary that the ancestral function of fibrinogen-like molecules is actually defense. So I would surmise that microbe and matrix recognition are common, complementary, ancestral properties of humoral pattern recognition molecules. And this is shown here in a schematic fashion: selected component of the extracellular matrix, selected component of the humoral arm of innate immune system, which can interact, and both are involved and can orchestrate tissue remodeling and repair, as well as antimicrobial resistance. We are very much interested in translating the molecule to the clinic. There is now genetic evidence, and I mentioned to you some new evidence linking polymorphism to susceptibility to Mycobacterium tuberculosis, to Pseudomonas aeruginosa. I showed uropathogenic E. coli and Aspergillus fumigatus. This is really work of Luigina Romani. And we are interested in translating the molecule as a candidate diagnostic and prognostic molecule. And the evidence suggests that it may represent an earlier marker better related to prognosis compared to C-reactive protein. In addition, we are moving towards an early clinical trial with a focus on Aspergillus fumigatus, which represents an important medical need. I would like to finish with, again, a general view of the humoral arm of innate immunity. Everything starts with the sensing of tissue damage and microbes, activation of the cellular arm of innate immunity, which is where all the hype is. But the very same cells, as well as other cell types, produce humoral pattern recognition molecules, with different tempos, and these are very heterogeneous in terms of molecular structure, including PTX3, the short pentraxins, ficolins, properdin, SAA, and you name it. But how do they work? They work by complement activation, opsonization, agglutination, which is more than an old-style immunological assay, is a strategy for disposal, agglutination, neutralization, and regulation of inflammation. This is the humoral fluid phase arm of the innate immune system, and of course, as I mentioned, there is a crosstalk between cellular and humoral innate immunity. And when I look at humoral adaptive immunity, antibodies, and PTX3 as a paradigm for the humoral arm of the innate immune system, well, of course, the structures are different, the cells that produce are different, the genetic strategy is different, but the functional outputs are the same: complement activation and regulation, opsonization, agglutination and neutralization, and glycosylation influenced regulation of inflammation. And it is my tenet that I share with you that these functional outputs are retained in evolution and have been potent drivers of evolution. I hope I did not forget to mention the people in the lab who have done the work. In particular, once more, Andrea Doni and Sébastien Jaillon, Barbara Bottazzi and Cecilia Garlanda, and many others, and all the collaborators that we have had over the years, and not listed here, and here is my office, and this is my role in the story. I'm writing or correcting the paper, and here is the PhD student or postdoc at the end of the project. And I thank you for your attention.